Hello everyone and welcome back to my channel. In this video I will talk about how can we partition the phenotypic variance of quantitative traits into different components. It is a very important topic of quantitative genetics and animal breeding. So I will try my best to explain it in a very simple and easy way so that you could understand it well. We will start this lecture by defining the phenotypic variation. Phenotypic variation can be defined as the observable or measurable differences among individuals within a population for a particular trait. So we can say all the differences which you can observe or measure among individuals within a population for a particular trait is known as phenotypic variation. And phenotypic variation is always measured in a population, so it is a population parameter. The term phenotypic variation cannot be used for an individual animal or individual person. It is always referred to a population. So in statistical terms, this variation or differences in a population are measured with the help of variance, and variance is defined as the average square deviation from the mean. So variance is also a population measure which is used to define the differences or variation in a population. That's why we sometimes also refer this phenotypic variation as phenotypic variance. So how can we partition this phenotypic variance of quantitative traits into different components? As you know that quantitative traits are those which are controlled by a large number of genes, and they are also influenced by the environment. So in mathematical equation, we can write the phenotype of a quantitative trait as P is equal to G plus E, where P is the phenotype, G is the genotype, and E is the environment. So the phenotype of a quantitative trait is a combination of both the genotypic effects or genetic effects and the environmental effects. Sometimes the phenotype of a quantitative trait can also be expressed as P is equal to G plus E plus GE, where G is the genotype by environment interaction. As some genotypes can perform different in different environment, so there might be an interaction between the genotype and environment. That's why sometimes we also consider this genotypic and environmental interaction, but it is not necessary, as it is, and it is sometimes ignored from the equation because it is very difficult to find out the genotype and environment interaction, so we ignore it mostly. And in this lecture I will also ignore this interaction to make the model very simple and easy to understand. So the phenotypic variance is denoted as sigma square p, and it arises due to genotypic variance and environmental variance. So we have just added sigma square in this equation. Sigma square p is equal to sigma square g plus sigma square e. So the variation or variance in the phenotype of quantitative trait arises due to variance in genotype and variance in environment. The genotypic variance can be further divided into three categories, which are additive genetic variance, which is denoted as sigma square A, and it can also be divided into dominance variance, which is denoted as sigma square D, and the genotypic variance can also be divided into epistatic variance, that is denoted as sigma square I. And the environmental variance can also be further divided into two types, which is permanent environmental variance or temporary environmental variance. But in most cases these environmental variances are considered in this sigma square E, and we don't partition this environmental variance into its components because it is very difficult to measure. So now our equation can become as sigma square p is equal to sigma square g plus sigma square e. So in other words, we can say the variance in phenotype arises due to variance genotype and the variance in environment. So now we will discuss the components of phenotypic variances in very detail. As I have already mentioned in previous slide that phenotypic variance arises due to genotypic variance and environmental variance. So now we will see what is the genotypic variance and what are its components and what are the meaning of the genotypic variance or environmental variances. This genotypic variance arises due to different type of gene interactions, and it is further divided into three categories, which is additive genetic variance, that is also sometimes known as genic variance. So additive genetic variance sometimes also referred as genic variance. The other type of genetic variance is dominance genetic variance, and the last type of genetic variance is epistatic variance. So now we will discuss all of these variances in very detail. So what is additive genetic variance? Actually, the additive genetic variance arises due to additive gene action. So the variance which is due to additive gene action is known as additive genetic variance. As you know that quantitative traits are controlled by a large number of genes, so each gene adds a specific value to the phenotype of a quantitative trait, and the resultant phenotype of a quantitative trait is the cumulative or additive effect of all of these genes. So this type of effect in which all of the genes adds a specific value to the phenotype is known as additive genetic effect, and this effect might be positive or negative. I will also provide an example of this additive genetic effect so that you could better understand this additive genetic variance. So here I will provide an hypothetical example of additive gene action. So consider four pair of genes which are capital A, capital B, small b, capital C, small c, and capital D, small d. So we have now four pairs of genes, which means we have 8 genes. And we will assume that each gene which is capital A, capital B, capital C or capital D, they adds 100 kg to the mature weight of cattle. So each of these genes, A adds 100 kg, B adds 100 kg, C adds 100 kg and D adds 100 kg to the mature weight of a cattle. But we will also assume that the genes which are small a, small b, small c or small d, they adds nothing to the weight, and the individual which has genotype small a, small b, small b, small c, small d will have only 800 kg body weight. So these genes they add nothing to the phenotype. So the individual which has all the small genes in the genotype will have 800 kg mature body weight. So in this genotype we have one capital A gene and all of others are small. So what would be the phenotype of this gene? The phenotype of this gene would be 800 plus 100. Where this 100 came? It came from the addition of this capital A, because all the capital genes they add 100 to the phenotype. So 800 plus 100 now becomes 900. So the individual which has genotype one capital gene and all other small genes would have the phenotype 900. And we are also assuming that there is no influence of environment. In this genotype we have two capital genes which are capital A and capital B. So in this case these two genes will add 200 to the phenotype. In this genotype we have 7 genes which are capital. So these 7 genes cumulatively add 700 to the phenotype. So the resultant phenotype of this individual would be 800 plus 700, that is equal to 1500. And in this case we have 4 capital genes, so they add 400 to the phenotype of this individual. So the resultant phenotype of this individual would be 800 plus 400, that is equal to 1200. So I am sure now you have understood what is additive gene action and the variation that arises due to this type of gene interaction, which is additive gene action, is known as additive genetic variance. So now we will discuss non-additive genetic variance. What is non-additive genetic variance? Actually, the dominance genetic variance and epistatic genetic variance are collectively called as non-additive genetic variance. So first we will discuss what is dominance genetic variance. Actually, the dominance genetic variance arises due to interaction between the alleles of the same gene, and this type of interaction is also known as intra-allelic interaction, and this interaction is mostly seen in heterozygous condition. So in heterozygous condition, one allele suppresses the effect of another allele, and this allele is known as dominant allele. The allele whose effect is suppressed is called recessive allele, and this phenomena or this principle is known as dominance. So in heterozygous condition, one allele suppresses the effect of another allele. That allele is known as dominant allele, and the allele whose effect is suppressed by the dominant allele is known as recessive allele, and this phenomena or this principle is known as dominance, and the variation which is caused by the dominance gene action is known as dominance genetic variance. So now we will discuss the epistatic genetic variance. So epistatic genetic variance arises due to interaction between different genes. In case of dominance genetic variance, I said that it was an interaction between the alleles of the same gene. But in case of epistasis, this interaction is not between the alleles of the same gene, but it is the interaction of between different genes. So we can define epistasis as the interaction between two or more genes where one gene masks the effect of another gene, and this gene is known as epistatic gene. The gene and the gene whose effect is masked is known as hypostatic gene, and this phenomena or this principle is known as epistasis, and the variation or the part of variance that is due to epistatic gene action is known as epistatic genetic variance. So now we will discuss the environmental variance. So what is environmental variance? Environmental variance can be defined as the phenotypic variance that is due to non-genetic factors. The variance that is due to non-genetic factors is known as environmental variance, and it arises due to feed, disease, management of farm, accident, or any other factor which is not the genetic factor which causes the variation. So the variation which is not due to the genes, the variation which is not due to the genes is known as environmental variation. So now we will discuss the permanent environmental variance. What is the permanent environmental variance? The variance that is common to repeated measures of an individual. The variance that is common to the repeated measures of an individual. For example, the beef cattle with only 3 teats. So the beef cattle who has only 3 teats, it will produce less milk, and the less milk will be available to its offspring. So the growth of all of its offspring will be compromised as less milk will be available to those individuals. So the part of variation that arises due to this type of environment which is permanent to the individuals. In this case, as I have already mentioned, that the beef cattle who has 3 teats will produce less milk, and less milk will be available to all of its offsprings. So this is a permanent source of variation in the performance of its offspring. and the performance of its offspring will be compromised. So the part of variation that is arises due to this type of environmental variance is known as permanent environmental variance. Now we will discuss the common environmental variance. First we will define what is common environment. Common environment can be defined as an environment that is shared with others during the development of an animal. The environment which is shared with others. So this type of environment is known as common environment. The performance of animals who shared a common environment will most likely be similar. So that is the simple thing. The animals who are raised in a same environment, a shared environment, will have uh, will perform most likely to be similar compared to those which are raised in different type of environment. But it will be different to those who shared another common environment. And the variance that is found between the group of animals who share different common environment is known as environmental variance. The example of environmental variance can be seen in the same litter group. The animals who produces more than one offspring in a litter, we can see the effect of common environmental variance in the lifetime of those animals. For example, the dam or mother is shared by the animals in the same litter group, and these animals also share the intrauterine environment, so their performance will most likely be similar. Performance of these litter group is also affected by the milk production of dam, milk composition of dam, and the mothering ability of dam. So the part of variation that arises due to common environment is known as in common environmental variance. So now as I have explained all of the components of variances in very detail, now we will see how can we add these components of variances into our model. In start, I explained the phenotypic variance model as sigma square p is equal to sigma square g plus sigma square e. Then we partitioned this sigma square g into three components, which were sigma square A, sigma square D, and sigma square I. And we also partitioned environmental variance into permanent environmental variance and common environmental variance. But again, as I have mentioned, these environmental variances are mostly ignored. So now we will add these components of variances into our model, and our model will now be extended like this. So the phenotypic variance is due to additive genetic variance, dominance genetic variance plus epistatic variance plus environmental variance. So now you can see I have replaced the genotypic variance with the components of genotypic variance. Likewise, you can also replace the environmental variance with the components of environmental variance, which are permanent environmental variance or common environmental variance. But as I have already mentioned that it is very difficult to partition environmental variance into its components, and most of the time these environmental variances are considered in this sigma square e, because in statistical term this sigma square e is also known as residual variance. So the part of variance that is due to unknown source of variation is considered in sigma square e, but it is possible that you could replace this sigma square e with the components of environmental variance. In that case, our formula will be extended as sigma square p is equal to sigma square A plus sigma square D plus sigma square I plus sigma square PE plus sigma square CE. But I have not extended this formula, and if you write this formula in this form, it will be correct. But if you want to partition the environmental variance, then it also depends on you. So as now we have seen that this phenotypic variance arises due to different components of genetic variance and the different components of environmental variance. But to actually know how much of the part of phenotypic variance is due to genotypic differences among the individuals or environmental differences among the individuals, we use a parameter that is known as heritability. So the heritability is defined as the proportion of the total phenotypic variation in a population that is due to genetic variation among individuals for a particular trait. So heritability tells us that how much of the total phenotypic variation in a population is due to genetic differences among the individuals of that population for a particular trait, and it is a proportion or it is a ratio. The formula of broad sense heritability is h square is equal to sigma square g divided by sigma square p. Actually, heritability has two types. One is broad sense heritability and the other one is narrow sense heritability. I will make another video in which I will explain the concept of broad sense and narrow sense heritability in very detail. But for this video, you have to know that heritability is a ratio between the phenotypic variance that is due to genotypic variance. And this type of heritability which is mentioned here is broad sense heritability, and the formula of broad sense heritability can also be extended as sigma square a sigma square d plus sigma square I divided by phenotypic variance. So we have replaced this genotypic variance with the components of genotypic variance, and we also replace the phenotypic variance with the components of phenotypic variance, and this type of heritability is known as broad sense heritability. So this was a lecture on the partitioning of phenotypic variance into different components. I hope now you can partition the phenotypic variance into its components, and now you have understand the concept of phenotypic variance of quantitative traits. So thank you for watching this video, and if you didn't subscribe to my channel, please do subscribe it.